Hello and welcome to this webinar titled Characterization of Biotherapeutics, hosted by Biopharma Asia magazine and presented by Diane Mccarthy, Senior Scientific Liaison at Global Biologics at the USP. This presentation will be followed by charge variant separation coupled to high-resolution mass spectrometry for routine MAB analysis, presented by Dr. Kai Scheffler, Product Manager at Thermo Fisher Scientific. My name is Stephen Edwards and I will be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now please allow me to introduce our first presenter, Dr. Diane Mccarthy. Dr. McCarthy is a Senior Manager of Science and Standards within USP's Global Biologics Department. Diane works with stakeholders to identify areas where standards are needed and define and develop new standards. Prior to joining USP, Dr. McCarthy was Senior Scientific Director at Caprion Biosciences, where she focused on the use of mass spectrometry for characterization of biologics and host cell proteins. Her previous roles also included Director of Scientific Affairs at EZO Sciences, where she focused on identification and quantitation of glycans by mass spectrometry, and Global Manager, Biomarker Research Center at Biorad Laboratories, where she directed translational and biomarker research contracts and collaborations with industry, key consortia, academia, and government groups. I'll now be handing over to our first presenter. Welcome, Dr. McCarthy.
Thank you, Stephen, for the nice introduction, and thanks to Biopharma and to all the attendees for inviting me to give this webinar today. So what I'd like to do is really give a broad overview of some of the aspects of characterization of biotherapeutics. So I'll start with a brief introduction to USP and what our role is in terms of developing standards. And then talk about different areas associated with characterization. So we'll talk a bit about CQAs, what some of the common CQAs are for biologics, and then I'll talk more specifically about size variance, charge variance, glycosylation, and a little bit on multi-attribute methods. The aspects I'll talk about, some of these will be compendial standards. Some of these are things that USP is currently working on, and so they're not currently compendial. But are things that we're hearing from our stakeholders are important and that we're looking to develop some standards for. So just to introduce USP briefly, USP will be 200 years old in the spring, and our mission has always been and continues to be to improve global public health. And there are a couple different ways that we address this. One is through our public standards. And we also have a number of other related programs. All of these are really focused on helping to ensure quality, safety, and efficacy of medicines and foods. We are a global organization, and so it may be a little bit difficult to see on this slide, but all of those little orange dots are locations where USP has either an office or a laboratory. And we currently have laboratories in the US, in Brazil, India, China, and Ghana. So our public standards come in two basic varieties. These are used in over 140 countries, and we have both what we call documentary standards and physical standards. So there's the Compendium, USPNF, that contains both monographs and general chapters. All of the monographs and general chapters that as they're developed or as they are revised, these are always published in the Pharmacopeia forum for public comment, and so they're open for 90 days for public comment. Many of the monographs and general chapters will have reference standards go through a multi-laboratory collaborative study before they are released as standards to define the quality of those or the numerical values for them. I should also mention that in addition to the monographs, the general chapters, there are two different types of general chapters. So general chapters numbered over 1,000 are general guidelines. They might have some aspects to consider and some general instructions, but they don't typically include a validated method. Whereas chapters that are under 1,000 are method chapters, they will have a validated method, and they often have reference standards associated with them. So I'll talk some about some of these chapters today, so I just wanted to mention that to start out with. Our standards are actually developed and balloted on by our Council of Experts. And so this is a collection of members, volunteers from industry, from regulatory bodies, and from academia and government. And these are the people that develop and approve all of our standards, whether those be documentary or physical reference standards. Within Biologics, we currently have 5 expert committees. One that focuses on peptides, focuses on 3 committee, which focuses on complex biologics like heparins and blood products. We have a group that is dedicated to antibiotics that have microbial assays. And then finally, we have a committee that works on general chapters. You might notice in this slide that this says 2015 to 2020 Council of Experts. So we work in a 5-year cycle. And so we are about to begin our new 5-year cycle in 2020. And the Council of Experts, or the collaborative groups, will change slightly. So in Biologics, in the next cycle, we will be introducing a new committee that will focus on advanced therapies, so for cell and gene therapy, as well as tissues and genome editing. And the other committees are fairly similar to what they are in the current 5-year cycle. So I'd like to jump in now to talk more about critical quality attributes. And I wanted to start with this definition from ICH Q8R2. A critical quality attribute is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. So I'll talk about today some things that can be critical quality attributes, but ultimately it depends on the molecule itself and whether these are important to ensure product quality. So these are things that need to be defined for particular molecules. We can give you some guidance in terms of what common CQAs might be. So I'm going to focus today on monoclonal antibodies just because this is the largest class of biologics currently. Many of the things that I'll talk about, though, are also going to be applicable to other types of biotherapeutics. And so when we think about CQAs, of course, there is the integrity of the molecule itself. If you start to see the SAB and the FC and lots of fragments, that would be an indication that the quality maybe is not where it should be. They need to be efficacious, so they need to bind to the antigen. They need to bind to the FC receptor if they're working through ADCC mechanisms, or to the complement interaction. In terms of physiochemical characteristics, anything that can change the nature of your monoclonal antibody could be a CQA. So we'll talk a bit about N-terminal heterogeneity, formation of pyroglutamate, any kind of amino acid substitutions or modifications like deamidation, oxidation, glycation, any fragmentation that might occur. Glycosylation is very important, in particular the fucosylation, sialation, and galactosylation tend to be, for many molecules, a critical quality attribute. The disulfide bonds, particularly if there's disulfide shuffling, that might change the structure and therefore the function of the molecule. And C-terminal heterogeneity, in particular, this tends to be lysine clipping. So I've summarized here some of the common CQAs and some of the test methods that are typically used to look at them. So for aggregation, the typical test method tends to be size exclusion chromatography that can be coupled either with UV detection or with multi-angle light scattering. Some orthogonal methods that are also used could be gel electrophoresis as well as analytical ultracentrifugation. For glycosylation, we're looking really at the presence of fucose, sialic acid, high nanostructures, and galactose typically. And these can be analyzed on multiple different levels. You can look at them as a released oligosaccharide or at the monosaccharide level. That's typically done by HPLC or capillary electrophoresis. Or you can also look at them at the intact protein or at the glycopeptide level, and that analysis would typically be done by mass spectrometry. Charge variants can include things like C-terminal lysine, deamidation, pyroglutamate formation, and these are often looked at by ion exchange chromatography, isoelectrofocusing, and to get to really a detailed level of understanding where the modification is, they're often looked at the, with peptide mapping and mass spectrometry detection. Oxidation, particularly on methionines, is a concern and can also be looked at by peptide mapping. And then the last two items on this list I won't talk about today specifically, but process-related impurities would include things like host cell protein, host cell DNA, residual protein A. There are a number of ways of looking at this. And then binding to the target or to the Fc receptor. That's typically done using ELISA, surface plasmon resonance, and specific bioassays. So I'll start out with size exclusion chromatography. SEC is a very robust method that allows us to resolve monomers versus higher molecular species, which can include dimers, trimers, and larger aggregates. It can also be coupled to multiple angle light scattering to get a size of the particular species. This is one area where USP does have compendial standards, so this is addressed in Chapter 129, Analytical Procedures for Recombinant Therapeutic Monoclonal Antibodies. And this chapter includes a validated method for size exclusion chromatography, as well as reference to a physical standard, the USP monoclonal IgG system suitability standard. So on this slide, I show some of the results that you might expect to get based on using this method. This is the HPLC chromatogram of the USP reference standard. And this standard is actually used throughout the run to bracket your sample solutions to make sure that the performance of the column remains consistent throughout the run. It's also used to define system suitability. And there really are three main criteria for system suitability. The chromatographic similarity, so you would expect to see a chromatogram very similar to the one shown here. The consistency of that chromatogram throughout the run, make sure that you don't see degradation of this profile as you get later in the run. And then the percent peak areas in the bracketing injections. And these peak areas would have been defined based on a multi-laboratory study to assess what was an appropriate range. And so here you can see you get very nice resolution of the monomer versus the low molecular weight species. We can see a very clear dimer peak just to the left of the main peak, and then some larger high molecular weight species as well. So I'll also touch on charge variance, and this is something I know Kai is going to talk about in more depth, so I'll just talk about it very briefly. Charge variants are often a CQA, and they do tend to be stability indicating. These can arise from a number of different processes, deamidation, formation of pyroglutamate, lysine clipping at the C-terminus, as well as any amino acid substitutions. And these are typically analyzed using either capillary electrophoresis or cation exchange chromatography. And on the right of this slide, I show you the profile of a monoclonal antibody standard that we are currently developing. And so we've assessed this material in cation exchange chromatography using both a pH gradient and a salt gradient. This is one of three monoclonal antibody standards we're currently developing, and this has a really nice charge variant profile because you can resolve at least 4 different acidic and basic peaks in this particular molecule. So it's very nice from the standpoint of assessing your method and assessing the resolution of that method. The next slide shows capillary isoelectric focusing on, I believe this is the same molecule. So the top spectrum on the left is a standard capillary IEF. So you see the basic peaks to the left and the acidic peaks to the right. We've also analyzed this molecule on imaging capillary electrophoresis, and one thing to keep in mind as you switch between those technologies is that the position of your basic and acidic peaks will switch. So now the acidic peaks are on the left and the basic peaks are on the right. But you can see, again, this molecule has a very nice profile in terms of being able to resolve the acidic and the basic variance. We also took this molecule and ran it through a stability study. So we looked at up to 4 weeks at room temperature, 37 degrees and 50 degrees. And so you see a selection of those CIF traces on the right. So we can see at room temperature, even at 4 weeks, we have very little change in the molecule. We start to see maybe a little bit of change at 37 degrees for 2 weeks, but by 4 weeks at 37 and certainly at 50 degrees, we see a significant degradation of the molecule. So this shows how you could use this to indicate stability. So I'll spend a little bit of time on glycosylation. Glycosylation is a major concern, in part because it's so complex and variable. Because protein glycosylation is not template directed, it really requires the interaction of a number of different enzymes and substrates. And so we have glycosylases, glucosidases, a number of different enzymes that are impacting glycosylation. It also can be impacted by factors in the cell line itself and in the culture conditions. So for example, the presence of manganese can be very important for getting the proper glycosylation of your molecule. The glycosylation of the final product can also be impacted by the purification process. So as you're going through process development, it's important to keep in mind that each of those processes may select a specific glycoform. And so it's important to be monitoring those during process development as well as at the final product level. And because glycosylation is so complex and variable, there is a possibility of having much more variability from batch to batch or anytime you make a manufacturing change, and also between biosimilar products. I bring this up just to show one of the nomenclatures for glycans. There are many different nomenclatures that people use. This is the one that I tend to use, and I wanted to point out a couple of different things. So one is that the different monosaccharide units have different color and shapes. We're particularly concerned typically about fucose, which is there at the bottom on the left-hand side. We're also concerned about anything that has a non-human glycan. So this includes N-glycosylaminic acid. This can be found in products that are produced in mouse cells. It's not frequently found in CHO cells, although I have seen it occasionally. And it has potential to be immunogenic. Also, the galactose, alpha-1,3 galactose motif is also a non-human structure and can be immunogenic, so this is something we want to avoid. So USP does have some compendial standards for glycoprotein analysis. This is a very complex graphic from Chapter 1084, which is the overview chapter in terms of different strategies to look at glycoproteins. And I've circled in here the ones that I'm going to talk briefly about in the following slides. But here I want to really point out that glycoprotein analysis can be done at multiple different levels. So you can look at the glycoprotein from the intact level, and this is typically done by mass spectrometry. You can release the glycans from the biotherapeutic, either through enzymatic means or through chemical means, and then analyze the cleaved glycans. You can cleave the glycans down to the monosaccharide level, which is often done to look at sialic acid. It's shown at the bottom. Or you can also look at them at the glycopeptide level. This is, I think, an emerging, so I'll talk a bit more spectrometry. So I'm just going to give a quick overview of the compendial methods for glycan analysis. I'm not going to go into these in any great level of detail because they're already laid out very nicely in these chapters. So these three chapters do contain validated methods for glycans. Chapter 212, which focuses on the released oligosaccharides, has an HPLC-based, multiple HPLC-based methods using either HILIC or HPAC, and a labeling procedure for 2AB labeling. There's also an option to use capillary electrophoresis, and this method also includes the labeling with APTS. Chapter 129, which is the chapter that focuses on monoclonal antibodies, also has both an HPLC-based method and a capillary electrophoresis-based method. The HPLC-based method here was actually developed before we developed Chapter 212, and so this has a labeling with a 2AA rather than 2AB. And then Chapter 210 looks at the glycans at the monosaccharide level. This is really focused on quantitation of sialic acid, so N-acetyl-neurominic acid or N-glycosyl-neurominic acid. There are two different release methods and two different chromatographic methods, along with the labeling procedure for DMB where required. We have several different standards, physical standards for oligosaccharides, and we've done some additional work on these to identify additional glycans. So if you looked today at the certificate that is in the USP store, the certificate for the oligosaccharide system suitability mixture A contains 8 major glycans. These are glycans from a polyclonal antibody. And we've done some additional work by mass spectrometry to identify now 30 peaks that correspond to 32 different glycan isoforms. And these are shown here. We also have some posters that we've presented at meetings that outline these results. And we've done the same thing with the oligosaccharide system suitability mixture B, which is primarily high mannose glycans. The certificate originally identified 5 glycans. We've now expanded that to 12 peaks corresponding to 14 different glycan isoforms. So now I want to go into some things that are not compendial standards, but they're things that we are currently thinking about and working on some standards for. So this is, I mentioned that you can look at glycans based on intact mass. And so this is taking 2 different monoclonal antibodies, measuring the intact mass, and then taking the deconvoluted mass spectra and identifying what glycoforms are there. So because there's a single glycosylation site on the heavy chain for a monoclonal antibody, and of course there are 2 heavy chains, we're always expecting to see 2 glycans. So for the spectrum on the top, you can see that the predominant glycans are G0F and G1F. So we have the first peak is 2 G0Fs, the next peak is 1 G0F and 1 G1F. We have 2 G1Fs and a little bit of G2F. The second antibody, the spectrum on the bottom, has a similar profile in the sense that we see primarily G0F and G1F in that monoclonal antibody as well, but we do see the presence of a new structure not seen in the other one, which is a G0F that has lost the glucanat, the little blue square. So this is one way to understand what's in your molecule. The other way that people are approaching this is at the glycopeptide level, and this is frequently now being done using multi-attribute methods. So this type of method really leverages the specificity of mass spectrometry and allows you to monitor a number of different CQAs in a single assay. This is something that I know a number of companies are exploring. As far as I know at this point, there is only one company that is using this as a release assay, but it is used quite frequently for characterization. And this is something that USP is exploring as a way for us to provide more efficient and comprehensive characterization of our own reference standards, as well as potentially whether there's an opportunity for a standard that would be useful in this area. And so we've been currently evaluating these 3 new monoclonal antibody materials that we're developing using this approach. The right side of this slide has a list of some of the common quality attributes that could be monitored in this way. I'll focus really on the top 5 of these. And so this is some results that we have comparing across 2 different monoclonal antibodies, a number of different CQAs. So you can see that there are some differences in these 2 antibodies in terms of the level of pyroglutamate. MAB-A has very little pyroglutamate, whereas MAB-B is 100% converted to pyroglutamate. They both have significant levels of lysine deletion at the C-terminus. When I looked at oxidation and deamidation, it's hard to compare directly across molecules because, of course, they have different sequences. But I picked a couple of peptides that were common across both of those. These are typically going to be peptides that are in the heavy chain region. And so you can see there is some difference in terms of the level of methionine oxidation between the 2 molecules. The deamidation, though, seems to be pretty consistent across both of these molecules. We were also able to look at the percent of total glycan. This is looking at the glycan at the intact glycopeptide level. And you can see that this really correlates with what we saw at the intact molecule level, where we have primarily G0F and G1F with a little bit of G2F. And the major difference you see between these 2 are the presence of the G0F minus N, which is minus the glucanac, or the non-fucosylated G0F minus the glucanac. We see a little bit of that in MAB-A, not so much in MAB-B. So finally, I'd like to wrap up with just some additional compendial resources. I think I've talked about the chapters that are related to aggregation and to glycosylation. In terms of resources for some of the mass spectrometry-based methods for looking at C-terminal lysine deamidation and oxidation, Chapter 1055 focuses on peptide mapping. This is a useful reference, as well as Chapter 1736 on applications for mass spectrometry. And process-related impurities, although I did not talk about that specifically today, if you're interested in particularly in residual DNA or residual host cell protein, we have some general chapters associated with that. And we do have a relatively new chapter, Chapter 509, which is a method chapter that contains a validated qPCR method for testing residual DNA. And that also has a reference standard associated with it, actually 2 reference standards, one for CHO and one for E. coli. And finally, I'd just like to wrap up. As I mentioned before, all of the standard setting processes within USP are really done by our expert committees. We are currently looking for candidates to join us for our 2020 and 2025 cycle. And so if you're interested in getting involved in the standard setting activity, I would certainly encourage you to take a look at this website and apply. Applications, I believe, are going to be open until May, and we hope that you'll join us. And thank you for your time and attention, and I'll pass it back to Stephen at this point.
Thank you, Dr. McCarthy. Now, before I introduce our next presenter, Dr. Scheffler, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the presentations. Now, please allow me to introduce our second speaker. Dr. Kai Scheffler is a chemist by training, holding a PhD in biochemistry. He is focused on the characterization of carbohydrates, peptides, and proteins throughout his entire professional career. Out of the variety of chemical, biochemical, microbiological, and analytical methods and techniques and technologies he has applied, mass spectrometry became one he got most fascinated by and thus has continuously deepened his skills and experience over the course of the past 22 years. He joined Thermo Fisher Scientific in 2004, 1 year before the first commercially available Orbitrap-based mass spectrometer was introduced onto the market. As support specialist for the proteomics market at first and biopharma a few years later, Kai has educated and supported users on the different Orbitrap mass spectrometer generations that were introduced in the past 15 years. He developed many relevant LCMS applications for the analysis of proteins and peptides, and in particular, antibodies and antibody-drug conjugates on the intact, subunit, and peptide levels. In his current role as product manager, he is engaged in fruitful collaborations aiming to further develop analytical LCMS methods and technologies, matching the advancing needs and requirements of the biopharma market. I will now be handing over to our second presenter. Welcome, Dr. Scheffler.
Thank you very much, Stephen, for the kind introduction. And also thanks a lot to Bright Talk for the invitation to support and complement today's webinar. My presentation is divided in a couple of sections, and I would like to give you a brief overview here. So I started with a very brief intro and workflow overview. We'll talk a little bit about sample preparation, then provide details on the setup of the LCMS system that also relates to buffer systems and columns. And then I will show you a number of examples that also relates then to data processing and reporting. So let's start with a brief introduction, which I can keep very brief because Diane nicely talked about a very similar slide already, introducing all the possible modifications that you typically encounter with monoclonal antibodies. And in charge variant analysis, the focus really is on all those modifications that do change the charge of the entire protein. So looking at the entire workflow, there are 4 key technologies involved. On one hand, the columns are really important that go hand in hand with the buffer systems, and they support the chromatographic separation of the charge variants. That's certainly also complemented by the LC system, and then moves on to the mass spectrometry system, and the data that I will show today are all acquired on a Q-Exactive biopharma instrument, but we also have recently launched a new instrument, the Explorers 480, that is equally suitable for this type of analysis. And the 4th key technology, as always, or in most of the cases, is software that supports the data interpretation. So regarding sample preparation, there is, for charge variant analysis, actually not too much to talk about. But that very much, of course, depends on where exactly the sample comes from that you desire to analyze. If it's, for example, a sample directly drawn from a fermenter, there are the usual purification procedures required, like fractionation and affinity purification. But if, for example, you would like to analyze the shelf life of a sample, a drug sample that's out of the bottle from, let's say, a pharmacy, there's absolutely no sample preparation required. You could directly pull it from the vial and inject it into the column. However, that's only when you desire to do the analysis on the intact protein level. There is, of course, also the opportunity to do that on the subunit level, which would then require an enzymatic digestion, for example, with IDS as one of the examples. However, in my eyes, it's beneficial to perform the analysis on the intact protein level because it will keep the complexity to a minimum and will not add yet another level of complexity, which would be the case if you would perform an enzymatic digestion step. So once the sample is more or less ready right away, or maybe after some level of purification, the next thing to consider is the choice of the column and the buffer system. And there are a few things that we need to review here when we talk about ion exchange protein separation. There are 2 general mechanisms to retain proteins on or elute them from what IEC columns. So on one hand, we have the more traditional salt gradient, and then on the other hand, the newer emerging pH gradient. In the traditional salt gradient-based ion exchange chromatography, the buffer, rather the pH of the buffer of the system is fixed, and it must be controlled throughout the entire analysis. The principle is that the proteins are eluted by increasing the salt concentration of the buffer, and that increases the competition between the buffer ions and the proteins for the charge groups on the column resin. And in this way, the interaction between the resin and the protein is reduced, and that's then causing the proteins to elute. So that's the principle for salt gradient elution. But then on the other hand, for pH gradient-based chromatography, the pH of the starting buffer is kept in a way that the proteins obtain the opposite charge of the stationary phase, and that way they attract each other and the proteins bind to their stationary phase. And then the proteins are eluted by changing the buffer, so that's typically increased, and the protein transitions then to a net zero charge, and in that way it's then eluted from the column. One of the benefits of pH gradients typically is that the salt concentration can be kept quite low, and that's yielding less buffer interferences. And also there are two ways of preparing buffers, so many different salts available. So on one hand, there are the non-volatile salts, and then there are the volatile salts. And I'll come back to that in just a second. But before that, there's just one comment also on regulatory requirements. Charge variant analysis is one of the requirements from government agencies for analysis of released products, because if a variation or variant occurs, means that a modification happened maybe during manufacturing already or upon storage, and that could be a big impact on the patient. So that's why, it's a regulatory requirement to perform this type of analysis. Now, when we look and try to prepare the analysis, the column choice is really important, and there are different columns available, which very much then depend on the PI of the protein. Different proteins have different PIs. However, antibodies all more or less have a very similar PI, above 7 or 8 even. So if the PI is below 6, then we have the ProPack SAX, the strong anion exchange columns available. If the PI of the protein is around 6 or maybe a little bit lower, then the ProPack Elite weak cation exchange columns would be suitable. And then for antibodies, which, as I said, have in most cases a PI above 6, the MAPAC SCX10, the strong cation exchange columns, will be suitable. And that's actually the column that we have used in the experiments that I show you data on today. Moving forward. So let's discuss the setup of the LCMS system a little bit more in detail. And there are the 3 key components I already mentioned. On one hand, it's the column, and very importantly, the buffer system, and also the MS system. And all of these key components require considerations regarding all parameters. Because, as I said, the buffers are critical, and the buffer that I would like to present here, the data with, they are all volatile buffers, and it's important that the pH covers a certain pH range to ensure proper elution. That very much goes hand in hand with the column and the entire system, the buffer and the MS. They also need to be correlated because, as I said, salt gradients, they are more critical when it comes to MS detection. So all these components need to be seen rather as an entire system and need to be adapted in a way that everything works well together. So I would like to give you one example of column length. I didn't mention in the previous slide, it did mention that we have used the 50 millimeter column, which is rather short. And just to show you the reason why this is actually a very well suitable column, this slide shows a nice comparison of 3 different columns that just vary in length. They're all 4 millimeters in diameter, but the top chromatogram shows 50 millimeter, the middle one 150, and the bottom chromatogram shows 250 millimeter in length. And the chromatograms from the elution profile point all look the same. Same pattern and same resolution, it's just shifted when we are using a longer column. So that shows that very short columns, like 50 millimeters, are absolutely suitable and even have the advantage of shorter gradients that can be applied and also shorter equilibration times that are required. Now, talking a little bit more about the pHs and the pH range, When we look at volatile salts, because what we were aiming to use and develop is a method that uses a pH gradient based on volatile buffers and salts that are MS compatible. But when you look at all the salts that are available, all the volatiles, there is, and that's a potential issue. But it can be overcome, and I'll show you that on the next slide. Before I move on, I would like to just mention the details regarding the buffers we have used. So for buffer A, we used ammonium bicarbonate, 25 millimolar, mixed together with 30 millimolar acetic acid, and that comes out to a pH of 5.3. And in buffer B, we've used 10 millimolar ammonium hydroxide. That comes out to a pH of 10.2. And then we used that short column, 50 millimeter in length and 2 millimeter in diameter, the MAPAC SCX column with a flow rate of 400 microliters per minute. And very importantly, I would like to mention that it's advised to use a pH meter to follow the pH real time, especially during the method optimization and when you try to develop a method and when you run samples for the first time. So this is now on volatile buffers, and as I said before, it has this issue of this buffering gap. When we look at salt gradients that can also be used to create a pH gradient, this is shown here, but this is non-volatile salts. We can nicely create a pH trace that is going in step, just to show you that the calculated and the measured pH really nicely match. That's shown in the little table on the right columns. So the calculation and the measurement work perfectly well, and a very nice pH trace can be obtained. However, the limitation of salt-based pH gradient is, in particular the non-volatile ones, is that they are not MS compatible. And that's why we decided to move on with the volatile salts. Again, they have this issue of the pH gap between pH 7 and 8. However, and I would like to move on to show you a real example, and this is cetuximab, showing you the chromatogram with a blue trace here. That's UV chromatogram with a detection at 280 nanometers. And then the gradient trace in this brown dotted line, and then the monitored pH trace line. This gap in the pH range 7 to 8 can nicely be overcome, and a very nice separation can be achieved. And cetuximab is a very nice example because all these variants do elude right in this buffer gap between 7 and 8. However, if the gradient is optimized in a way that it's really shallow, still a very nice chromatographic separation can be obtained. So now moving on to mass spec detection, because the previous slide was UV detection as mentioned. But if we want to hyphenate now the LC with MS, there are a few things to consider and to know up front. So I would like to briefly explain the difference between denatured and native conditions that the proteins are analyzed with mass spec detection. Under denatured conditions, the protein is exposed to organic solvents, to low pH, and they unfold, they are being denatured, and they expose a larger surface area. So we can attach a lot more protons, and that results in very high charge states, and many of them, which we see on the top left chromatogram, or rather MS spectrum, that shows many, many different charge states, very nice Gaussian-shaped envelope, and it typically ranges between 30 and 60. When we run the very same protein under native conditions, so we expose them not to any organic solvents and we keep a near neutral pH, we are using volatile salts, then the proteins are kept more happily. They don't unfold, they keep their three-dimensional structures. That means they cannot attach or accept so many protons. As a result, on the mass spec level, we see lower charge states and also fewer of them. So when we detect lower charge states, we see a shift on the M/Z scale, on the x-axis, to higher M/Z levels. And on the lower trace, where it says native, you see a very narrow charge envelope. So that also shows you that detection capability beyond the master charge 6,000 is required. And that is really important when we look at some more specific settings of the mass spectrometer, and here are shown details for the Q-Exactive Biopharma that we've used in the experiments. And there are, on one hand, a few settings that relate to the ion source. On one hand, the capillary temperature, which should be kept as low as possible to maintain the protein in the native structure. And also, the gases in the ion source are really relevant to ensure good dissolvation, also stability of the spray, and a very good sensitivity. And when we look at the mass range that's required, the Q-Exactive Biopharma offers the so-called HMR mode, that stands for high mass range, and that opens up the detection capability beyond 6,000 all the way to 8,000, which is perfectly suitable for analysis under native conditions. And you see that also on the line in the second from the bottom, the scan range 2.5 and 8,000. That's what we set it for because the HMR mode allowed us to set that. Also, and I think that's even the most critical parameter, is the source CID or in-source CID. That's really important for dissolution, for declustering, and also for removing salt adducts. And most typically, the lower settings will not work, so one needs to tend to go to the higher limits. Not setting that at all will result in very bad spectral quality, so this is really important to remember this setting to keep this high. The resolution setting very much depends on the size of the protein and also what the desired outcome is, if isotopic resolution can be achieved and should be achieved. For monoclonal antibodies, as they are very large in size, typically in the ballpark 150 kilodaltons, it makes only sense to set the resolution setting to 17,500 or 35,000. Gives the best sensitivity and will provide perfect resolution to resolve the different glycoforms, but as I said, it cannot achieve isotopic resolution anyways. Okay, I would like to now move on and show you some examples. So you see on the left side 7 traces of different antibodies we have analyzed with the volatile salt-based pH gradient and the column already mentioned. And you see also that the different PIs are listed next to the names. On the left side, all these runs were created with just a generic, very simple, non-optimized gradient running from 0 to 100% B. And you see it works very well for all the samples. However, the resolution of the separation of the charge variance can be improved. And that was done in a second round analysis. So for every antibody, as they all have slightly different PIs, the gradient was adapted and optimized. And then for every one of these 7 antibodies, we got the best resolution in the chromatograms shown on the right side. I would like to zoom in the next slide in the picture of or chromatogram of trastuzumab that's boxed here in yellow. From the distance on this slide, it looks like, oh, there's one major variant and a small peak, maybe not a lot more. But once we look at that in more detail, we see, again, the blue trace is the UV chromatogram detected at 280 nanometers. And the measured pH trace shown in black and the gradient in brown dotted line. Now looking at the blue chromatogram, you see with a closer look, there's actually a lot more going on. They're small intensity, but very nicely detectable variants. So that's on the UV level. And if we take it one step forward, including the MS, now we see the MS-based chromatogram in the bit of the mass spectrum, representing all the different charge states of the main variant that alludes in this main peak at 7.7 minutes. On the right side in the small panel, we see the results after deconvolution with the assignment of the glycoforms, and the table above shows you the theoretical and experimental values with very great mass accuracies. So this is just to highlight in one picture here the spectral quality that can be obtained using this workflow I described with the column, with the buffer system, with the LC, and with the MS. Now, I've shown you this slide before with the 7 different chromatograms, and I just wanted to pull this back up to let you know that in the next couple of slides, I would like to give you more details on this particular antibody box we analyzed in much more detail. And looking into that, in a little bit enlarged view, you can also see here there are actually three rather main glycoforms. One is really outstanding, then we have a one in the middle or close to 8 minutes and a small one to the right. And there's a lot more peaks that can be detected, especially with an enlarged view. There are actually 9 acidic variants and 6 basic variants that could all be identified. Of course, not by just reading and looking at the chromatogram. Now this is really entailing now the mass spectrometry that provides us with all the data, and then also the data analysis and the software support that we need for analyzing all these data. And that brings me to the last point. I briefly just want to mention a few things on the data processing level. There are 2 software packages available. On one hand, this is Biopharma Finder, and on the right side, you see the Compendium CDS software, both of which can perform the protein deconvolution of intact protein spectra very nicely. Both use the same algorithm called respect and extract. However, the benefit of using Compendium is that it offers compliance, and that relates, for example, to audit trails and data integrity and anything you wish for in regards to compliance. When we look at how we perform the protein deconvolution in either one of these 2 software packages, there are 2 different ways how this can be done. And that very much depends on the level of information that's available already for the sample. So if you run a sample for the first time, you don't know what to expect, you don't know what masses the protein may have or what variants you may see, you may want to go for the non-targeted approach. So you will just run through the software with your raw data, and you will get a list of masses that are graded upon the deconvolution. However, if you know already what to expect, you would like to confirm certain variants, you would like to see that it's the same peaks and masses you've already analyzed in a previous analysis, you could choose the target of masses you expect, and then the software will try to match all the masses from your new sample with the mass table that you provided. In either way, whether you go the non-targeted or the targeted approach, there's a very nice feature available that's shown here in the middle blue box on the bottom. It's called the sliding window feature. And I would like to explain that a little bit more in detail here on the next slide. On the top left corner, you see a chromatogram, and it's again the adalimumab chromatogram with the 3 main peaks, and you may recognize also the smaller peaks. But what I would like to point out here, I've put a lot of these orange colored boxes across the chromatogram, and they represent the sliding windows, as the name of the algorithm also is. So these windows represent very narrow retention time windows. You can define the width and also the level and extent of the overlap of these windows. However, the overlap is really important, so you don't miss. And I'm going to go through the entire chromatogram step by step, and we'll take all the data that are within one window, does one deconvolution step, and we'll create one spectrum per window, and keep all these individual spectra in the memory. And once you've come to the very last step, the last window, it will take all the spectra together and will create one combined result, one spectrum that you get as a user. So you don't have to deal with all these zillion windows or spectra, but just a single result output as a spectrum. The benefit of this is that all the peaks in terms of their relative intensity reflect the intensities of your species across your entire run. So this is particularly important for the very small and low abundant species, because if you did the alternative approach and you just took your mouse, dragged across the chromatogram, averaged the entire run, and then just did a single deconvolution step, all the very small species would get discriminated and lost potentially. And that would mean a lot loss of information from your sample. So this is just a brief introduction into this feature, which we have applied throughout the analysis. And just to give you an idea, this is showing a table. This is just part of the results, and I will not go through all the numbers. But these data show all the results from triplicate runs. So this sample was run in triplicates to also ensure the reproducibility of the analysis. And it mainly shows all the lysine truncations and also the different glycoforms that were nicely, reproducibly detected and analyzed throughout all these 3 bonds. So this is just one part of the charge variants that were detected. And then moving forward, that's still the same chromatogram. It just looks slightly different because the big peak is now a bit cut off and some of the peaks are boxed here, to just highlight a few things. So on one hand, on the top panel A, on the very left side, there is a very tiny peak that was identified as fragments. Then right from that, with the red arrows, we found some deamidations. Then the main peaks, they are all relating to lysine truncations. And there's also labeled here with the blue arrows the succinimide containing variants that could be detected. And also highlighting the very small spectrum on the very right side that has also the Glycan annotation, shows that with a mass spec you actually take your data analysis to a much higher level compared to UV detection. In UV detection, you can make out a number of small peaks, and all these 16 peaks I showed on the slide before, but really interpreting all these peaks and also the very low level peaks, this is where really the mass spectrometry setup brings all the data to a new level and provides a wealth of information. And the last data point I would like to show, and again, it's still the chromatogram, yet just differently colored representation here, shows again on the left side the peaks 1, 2, 3 boxed in red are deamidations, and there are also glycation variants found here, shown these peaks that have always shoulder to the left of the main lysine variants. They were also nicely identified in all the triplicate runs. That's also shown briefly here in the summary table. So in summary, we found many different variants for this one sample that was run in triplicate runs, and that already brings me to my conclusions. I hope I was able to show you the entire workflow, starting with the column discussing the buffer system briefly, also the chromatography, the pH gradient, the volatile salts, then the mass spectrometry, the data in different levels, and how it brings the accurate masses very high confidence results, enabling us to interpret all the data and confidently annotating the entire chromatogram, finding many different charge variants from lysine truncation, glycan profile, oxidation, deamidation, glycation, and also succinimide formation. And the beauty is that it's also possible, based on the data, to not only identify peaks, but also to perform a relative quantitation. So with that, I would like to acknowledge a number of collaborators and also colleagues, and I'd like to start with Jonathan Bones and his very talented and skilled and very productive team at the Nyberg Institute that's located in Dublin in Ireland, and also my colleagues from Thermo. It's a long list. And from all these people, there are in particular 2 colleagues I would like to mention, namely, on one hand, it's Florian Füssel from the Nyberg Institute. I think he's become a master in chromatography for pH and charge variant analysis, and also for native MS analysis. And also Ken Cook, he is in Thermo the guru for biochromatography. And both these colleagues I just mentioned provided most of the data that I present today. But again, thanks to everybody that's listed on the slide, including our chameleon mascot, Charlie, and his variants. And I would like to conclude by thanking you for your time and your attention, and I hand back to Stephen.
Thank you, Dr. Scheffler. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, can other methods be used other than what is included in 129, for example, the ICE or SEC by UPLC? That question is for Dr. McCarthy.
Thanks, Stephen. Yeah, so the methods in 129 are validated methods. They can be used, but there is always the possibility support any method in USP that you can use other methods. I think it's General Chapter Section 630 says that you can use other methods when those are validated against the compendial method. I should also mention that these procedures that are in any general chapters are only required when they are referenced in the specific monograph. And so right now, that particular chapter is not referenced in any monograph. So these are they're really there to be able to help you to streamline development. If you can use them if you wish, you can also use other methods as well.
Thank you. Our next question is, this presentation focused on monoclonal antibodies, but how does this apply to other types of biotherapeutics? That question, again, is for Dr. McCarthy.
Sure. So I think I did show monoclonal antibodies as an example in this case, but many of these CQAs will apply to other biotherapeutics as well. Obviously, anytime you're looking at glycosylation, you might have a molecule like erythropoietin as an example that's heavily glycosylated. These methods will be relevant. The charge variants may be different. You may not have something like the C-terminal lysine clipping or pyroglutamate in that particular case, but you could still have amino acid substitutions. You still would have deamidation, oxidation. I would say that, the basic methods are probably applicable. There may be more optimization needed to get the right chromatographic conditions for another molecule just because of differences in terms of size and charge.
Thank you. Moving on, our next question is, what type of UHPLC system do you recommend for using for CVA analysis, or rather, what requirements should it fulfill regarding retention, time stability, reproducibility, and pressure limit, and alike? And that question is for Dr. Scheffler.
Okay, thank you, Stephen. In our experiments, we've used the Vanquish Flex binary system, which delivers really great accuracy and precision and also sensitivity with a very low gradient delay for fast gradient separation. The pressure limit on this system is 1,000 bar, but that's definitely not required for charge variant analysis. Typically, for the data I've shown, the pressure we acquire or encounter is in the ballpark of 150 bars. When the column is in good condition, it may rise a little bit to, let's say, 300-400 bars when the column gets really old and gets close to needing a replacement. So regarding the pressure, any system that can handle 500 to 600 bars will be fine. But what's really relevant is the reproducibility when you are aiming for routine analysis and when you also rely on reproducible retention times. So the bottom line is that reproducibility and the generation of fast gradients are the key criteria that the LC system should really fulfill.
Thank you. Our next question is, what level of sensitivity does the workflow you described here provide relating to amount of sample injected and abundance level that can still be detected? That question is again for you, Dr. Scheffler.
Yeah, okay, thank you. So typically, and I haven't mentioned many details on the amount of sample that we've injected, but typically for CVA analysis, injections are in the ballpark of about 50 to 100 micrograms, which is quite a lot. It is possible to use less material in the ballpark of, let's say, 10 or 20 micrograms, but the key in this analysis is to really detect also the very low abundant species, and in this case, it's beneficial to use higher amounts. When we look at the entire range of species we can detect, regarding the lower limit in particular, in terms of relative amounts, it's possible to go down to levels definitely far below 1%, and typically even in the ballpark around 0.1% plus minus, it is possible to really reproducibly identify the species and also quantify them.
Thank you. Our next question is, have the charge variants detected in CIEF and CEX chromatography been identified? My question is for Dr. McCarthy.
So the data I showed is actually very recent data, and we are working to identify what those individual peaks are using both the HPLC to MS approach as well as the CE-MS approach. So we don't have that yet, but we're working on it.
Thank you. Moving on then, our next question is for Dr. Scheffler. You mentioned in one of your slides that CVA analysis is a regulatory requirement. So I'm wondering, what is your assessment on how easy, robust, and reliable this workflow is for implementation in a routine lab?
Yeah, that's a very good question. So the data that I've showed in the presentation, I mentioned on one hand that they do require some optimization unless you're happy with the separation that can be obtained just with a standard gradient. But it's actually not too much work to optimize the gradient for the individual samples. But again, it's the question of what exactly you aim for, but ideally I would say a little bit of optimization is needed for a sample that's not known. However, if you are running the same or very similar sample day in, day out, you can probably go with one optimized gradient and you don't have to adapt that every day. So that's just for the gradient adaptation. Regarding the rest of the setup, I think it's very easy and very robustly possible to set it up. And as I said, regarding the requirements for the LC, as long as this is very reproducible and you just aim for comparing samples, this works really nicely. And also when we look at the buffer systems that are entailed, the volatile pH buffers, you can prepare them as 5x stock solutions and you can store them for quite a while. However, it's actually advised that you prepare fresh dilutions of the buffer, if possible, about every day. But I mean, this is just a dilution step. And all in all, bottom line is it's working very robustly. There are a few things to consider and typically to maintain, but in a routine lab, once you've established that, you know how to optimize gradient with a little bit of work, that works very, very easily and very well.
Thank you. Our next question is, how can we collect innovator reference sample for comparability? And that question, Dr. Scheffler, would you like to answer this first?
I wonder whether actually Diane would be a better person to answer this question.
I can try to address that. I think this is something that we're aware of as being a challenge to the industry. It's probably not an issue that either Kai's organization or USP can address directly. I think this is an area, though, that we know that regulators are aware of the issue and are working to help resolve it. So I would, just suggest you talk to the local regulatory authority about that question.
Thank you. Our next question is, can HPLC or HPLC MALS is a good enough technique to identify higher order aggregates? Higher order aggregates may sit the column frit or block the pores of the column. Dr. Scheffler, would you like to answer that one? Or Diane?
I think that might be for me.
Yeah.
So, yeah, that's a good question. I mean, there certainly could be higher order aggregates that would be too big for HPLC mols. We've used it, quite a bit for monoclonal antibodies. It seems to work quite well. But I think this is where it's really important to have orthogonal techniques. So, something like the analytical ultracentrifugation might show you something that you don't see in SEC mols, or likewise even a gel, if you have something that's not entering a gel, that might be a good indication that you've got a really large aggregate. If the aggregates are very large, you might also be able to see them when you start to look at your subvisible particles, or even potentially, I guess, visible particles. So this is, again, orthogonal techniques are key here.
Thank you. What is the optimal pressure required for CEX and analysis of charge variance? And again, I believe that would be for you, Dr. McCarthy.
That one I don't know off the top of my head. I'd have to go back and look at the methods we use. But I think, there are so many different, columns and vendors here. I think it may be most advisable to go with, what's recommended for those particular columns. Kai, do you have any other comments on that one?
Yeah, I think the pressure is not all too critical, and I think it very much depends on the buffer system and the particular column and the flow rate you're using. So it all needs to be a little bit optimized together, but the pressure per se I wouldn't take really as an indicator. And as I briefly mentioned, in the setup we've used with a 50 millimeter column and 400 microliter flow rate, we have about 150 bars. If we would lower the flow rate, then the pressure would certainly be a bit lower. If you have a longer column, the pressure will also be different. So one can't really say that per se. It really depends on all the parameters, on the column length, the diameter, and the flow rate. But typically the pressure is not overly high, so it's not really a UHPLC type analysis. So as I mentioned, typically the pressure is in the range between 100 and 300 or 400 bars.
Thank you both for your answers there. Our next question is, does LC part separates the product first and automatically send the... who'd like to answer that first? Dr. Scheffler, should I answer that first?
Yeah, I can take that on. So that's the idea and the concept of doing an online analysis where the LC is directly connected to the MS. So whatever the LC separates will be directly sent on to the mass spectrometer. And the separation always needs to come first, and that's the idea, because if we just directly injected a sample without any separation, we would get some information, but we would not be able to really separate the variants, and in particular the low abundant variants. So yes, the LC does first the separation, and we don't have to pick and choose what we want to analyze with the mass spec. The entire chromatogram, in most cases we actually want to have the entire chromatogram be detected and evaluated by mass spec, and at the end, when the run is done, then we just look at all the data. Yeah, so it definitely, yes, it's automatically sent, and that's the idea of an online LCMS analysis.
Thank you. And with that, I will conclude the questions and answer session. Thank you, audience members, for your questions. Before we finish the webinar, I would like to ask our presenters, Dr. McCarthy and Dr. Scheffler, if you have any closing remarks for our audience. Dr. McCarthy, would you like to go first?
Sure. I'd just like to thank everybody for attending and encourage you, if you do have questions about any of the methods that we talked about, particularly some of the ones in the compendium, feel free to reach out to USP. The USPNF has a link to the appropriate person that works on those particular chapters or monographs, so you're always able to contact someone here with your questions.
Thank you. And Dr. Scheffler, do you have any closing remarks for our audience?
Yeah, I hope that we were able to give you a good overview on characterization of biotherapeutics, the different methods. I, in particular, dived into the charge variant analysis, and using the pH gradients with volatile buffers is a rather recent method. There are a number of publications, few ones, but very good ones out there. And I think it's a method that will find really wide applicability and interest. So I hope we were able to encourage you to look into that in more detail and try it. And as I said, it's definitely has potential to become a good method even in routine labs. If you feel like there's any further information needed, I can encourage you to check the Thermo website. There are already a number of application notes out there, or please feel to reach out to me or any of our colleagues. Thank you.
And I would like to take this opportunity to thank our presenters. Once again, we had Diane Mccarthy, Senior Scientific Liaison, Global Biologics, a U.S. Product Manager at Thermo Fisher Scientific. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send in your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.